The cytoskeleton is a network of protein fibers that determines the shape of cells without cell walls, provides for the movement of cells, provides for the structural support and movement of organelles within the cell, and provides the framework for moving and separating chromosomes during cell division. The three types of protein fibers that make up the cytoskeleton are thick microtubules, thin microfilaments, and medium-sized intermediate filaments. Microtubules are hollow tubes assembled from subunits made out of the protein tubulin. Subunits not forming microtubules are dissolved and scattered throughout the cytoplasm. As subunits are readily available, microtubules can be assembled and disassembled as required by the cell. Subunits are assembled into microtubules by microtubule organizing centers found at various locations within the cell. All eukaryotic cells have a microtubule organizing center near the nucleus, called the centrosome. During cell division, the centrosome produces a football-shaped array of microtubules called the spindle apparatus that separates chromosomes and divides them between the two new daughter cells. In animal cells, a pair of organelles called centrioles are located by or in the centrosome. Centrioles are short, barrel-shaped rings with 9 microtubule triplets forming their outer edge. As a cell prepares to divide, the centrioles are duplicated, and one pair moves to each daughter cell. In cells that have structures called cilia and flagella, the centrioles multiply and move to the surface of the cell, where they become anchored just beneath the plasma membrane, forming basal bodies. 10 pairs of microtubules grow out of each basal body. One pair grows from the center of the basal body, while the other 9 pairs form the outer infrastructure of a cilia or flagellum. A gap the width of a microtubule exists between each of the outer microtubule pairs. Numerous motor proteins firmly attached to one pair of microtubules have legs that cross the gap and can walk on the adjacent microtubule pair. The force applied by the motor proteins causes the microtubules to bend. The synchronized movement of large columns of motor proteins attached to microtubule pairs trying to walk up or down adjacent pairs creates the rowing motion of cilia and the wave-like motion of flagellum. Cilia, Latin for eyelash, are short and numerous structures that extend from the cell membrane. Cilia move single-celled organisms such as paramecium through their environment with a rowing motion. Flagella, meaning whip in Latin, are usually considerably longer than cilia and fewer in number. Flagella move in a wave or whip-like motion. The sperm of most animals and algae rely on flagella for movement, as do a number of single-celled organisms such as euglenoids. Microtubules are also believed to be involved with the movement of organelles, such as vesicles, within the cytoplasm. One hypothesis holds that vesicles and other organelles have receptor proteins to which motor proteins attach and then walk an organelle to its destination via the network of microtubules that extends throughout the cytoplasm.